The overall goal of this in vivo matrix gel assay is to investigate the interaction of endothelial cells with pericytes.
This matrigel assay is designed to help investigators study the major steps in angiogenesis. It also will allow the identification of signaling pathways responsible for major steps in blood vessel formation, as well as genes whose dysfunction can lead to vascular disorders and other aberrations of angiogenesis.
The main advantage of this technique is that it allows us to study the in vivo blood vessel formation involving both endothelial cells and their support cells, pericytes.
To begin, grow human endothelial and pericyte cell cultures on 100 mm plates with 10 milliliters of the appropriate media. Change the media every 2 to 3 days until the cells reach 80% confluency. In order to prepare the matrix gel solution to mix with the cells, thaw a stock of matrix gel overnight at 4 degrees Celsius. Then make 1.5 milliliter aliquots for storage at -20 degrees Celsius long-term, or at 4 degrees Celsius for up to 2 weeks. Each experimental plug will require 200 microliters of matrix gel, so one stock tube can supply 6 plugs with a 25% excess. Prepare each required aliquot for use by adding 15 microliters of thawed 100x beta-FGF stock. Mix the beta-FGF into solution by pipetting, then incubate on ice for at least 30 minutes prior to adding cells.